In this session, I will talk about how to interplay with some isolated tools. The isolated tools means some tools that we don't incorporate or fully incorporate with Giottos. Today's tools I will introduce is some track, which is performing the spatial niche trajectory analysis. So for on track, uh, it's a niche-centered and machine learning method for calling these spatial continuity trajectories, which is based on the niche level features and smoothing GNN-based algorithm to create a probabilistic clustering result, and based on this clustering result to build this trajectory. And here it has one point to show some details here. So the first question is that why we need a spatial trajectory. It's because when previously we could have some spatial domain detection method give some output of spatial domain, but the question is that in this spatial domain there may be some different cells or spot here, but their environment may be very different. Take this 3 points as example. For this cell, they are surrounded by the organ and the dot, gray dots, and this one is also surrounding with orange dots, but for this one, it's almost surrounded by green dots. Obviously, they are located in different microenvironments, but if we are taking the spatial domain detection, then maybe cluster as inside of the same spatial domains, but actually they are different. So we need a continuous metric to describe these spatial variations. And the second question is that why we need a niche trajectory, not through time like what we are doing in single cell analysis. The question is that take these two cells as example, they are different cell types or from different lineages, but they are spatial neighborhoods. That means they should located or share a similar microenvironment. But if we are using the through time analysis, they will belong to different lineages. So their through time may be very different. That's why we need a niche trajectory. So, and the third question is that how could we construct a niche? As the niche is multicellular spatial localized regions where there may be different cell types coexist here and interact with each other. So we performing like this way. First, we select anchor cells, and the second, we are finding some nearest neighborhoods to these anchor cells, and last, we call this area as a niche, and we can iteratively to define the anchor cells to every cell. So we have many niches here. And the next question is that how could we calculate the features of this niche? And the solution is like that. First, for each niche, we have their cell type information. And second, based on the distance from the cells to the anchor cells distance, we perform normalizations. And after that, you can get a new representation for each niche, which is based on the cell types within this niche. And after that, we can build a niche network. The network between this network is the spatial network, and the edges between each niche is defined based on the anchor cells relationship. And in this GNN part, we will finally get a niche cluster. It's a cluster about each and the things should be noted that this is a probabilistic clustering method. That means each niche has a probability to assign to each niche clusters. And we can also get a connectivity matrix for each cluster. This connectivity is based on their spatial uh, spatial continuity. And during this training process, we will have using this loss function to control how the clustering results. And the first term is the modulity. The modulity means that for each niche cluster, it should be spatially continuous in our samples or slides. And the second term is the purity. That means within each niche clusters, there niche features should be same. And the third term is a regularization term to prevent that the situation that all the niche will assigned to the same niche clusters. And after that, we can build a niche trajectory based on the spatial relationship within each niche cluster and assign the values we call the NT score here to describing the spatial variations of these niche clusters. And after that, we can map back this niche NT niche trajectory scores to the niche level and to the cell level. And in the end, we will have a metric for all the cells about the spatial variations. So, and in today's case, we will apply on-track Murphy's data. So the Murphy's is one type of facial technology, which is image-based spatial transcriptomics technologies which can detect many cells in the same time. And in their last paper, they can detect like the whole mouse, whole brain data set with million cells. And okay, so before we download any data sets, let's create some database and result flow. So for today's data set, it's motor cortex data set. In this data set, they have 61 tissues, which contains about 218 cells with 258 genes panel. And in the original study, they have already classified cells into three main groups like glutamatic neurons, GABAergic neurons, and non-neuron cell types. And they can further divide these cells into 24 subclasses, subclass level cell types. And and their raw data sets could download here soon in this script, but as I already downloaded them, so let's see here. Yeah, I already downloaded, so because their data is a little bit larger, so I will skip this part, but no worry, if you want to follow me, you can. I have provide the processed data in this Zenodos, so no worry about that. So first things is that we need to create our Giotto object. As you have seen this data as this script, very similar script before, like we specify the instructions of this Giotto, and as there this Murphy dataset is not saved in a standard format, the expression information is stored in a data format, so we can read this data object files directly using the data tool, just like what Akra described before. And for metadata, we can just read the metadata. And one thing should be noted that in their metadata, their cell ID are all fixed numbers. So we should tell the R that all we should treat them all as character to avoid some limitations of such a long fixed numbers, and try to set the cell ID. So you can see the metadata looks like that. It's a cell ID with sample ID. It's like that. It's about the samples and class level subclass level. They have label levels cell type, but we won't use them. So here we only use two slides here to speed up because their total size is a large scale set. So after that, we can subset the Giotto by the cell ID to create two Giotto objects for the slice 1 and slice 2. So, and add the metadata to these two Giotto objects. And the third thing is that we need the spatial information as they not provide the spatial information directly. We need to infer the spatial information about for each cell through the cell segmentations. Like this one is we just use the median of their shapes as the x and y coordinate for each cells. And after that, we can merge these information together and subset the selected samples and create the spatial locations object from this X and Y with cell ID. And after that, we can assign this spatial location information to our Giotto object. And the last step is merge these two Giotto objects together, like what Jeff told us in this morning section. And after that, we can save this Giotto object. So yeah, if you, and actually, if you don't want to download such a large dataset, you can directly download the pre-processed data here. It's a quite small dataset, just you don't want to take such a long time. So this is the Giotto object we created. Also, next image crash if you, which you can here. So let's go back to the R and we can load the Giotto object back here. And to see what the data look like, you can see here, these are two samples here, and this is a cell type annotation information. And this to be mentioned that this is a mouse brain dataset, and the VLMC is annotated the boundary of this brain and going to the deeper layers. So there's exists a nature coordinates here that is from the boundary to these deep layers. The main idea for the spatial trajectory is build this nature metric. So and the next step is how we create the input of round track. This is already, we have a function here. So it will be already generated and saved to the disk. And we can see what the inputs look like. That's in the cell ID, the samples, and the X and Y coding and the type information. So as I told before, the on-track for now is still isolated to the tools. So we should run it separately. You can run either in your own laptop or HPC with GPU, because on-track is GNN-based method. It will be more quick if you run on GPU with large dataset. For this dataset, it will run about 6 minutes here, depending on which computer you are using. So for installing on track, you can use the command like that. So, okay, this is how we create a new environment with string counter and install on track. And for running step, the command is right here. You can run it, but it will take about 6 minutes. So let's just skip here for now. Can load it with the existing result. And we can see what results loaded in Giotto. So first thing is that the have niche clusters and niche core information in this metadata. And the second thing is that we have two metrics here. First is the probabilistic metric for each cell to each niche cluster. And the second thing is that it's the connectivity matrix for each cluster, which is stored in these two positions. And the first thing we want to know is that what the niche cluster looks like. And we can use the spatial feature plot function to look at them. This one is size small variable. You can see that for these two samples, both of them have this 6 niche clusters. That's what we define here. And to be noted that this is a probabilistic assignment. So you can see that in the center area, it's almost the population is almost to 1, and in the edge area, it's further away. And in this gray area means that for these cells, they are not assigned to niche cluster 0. So you can see that this niche cluster have a very clear band here to indicate the different features. And if you want to look at binarized versions, you can use these functions which is store the binarized niche cluster information in the metadata. And you can see that from the boundary of the brain that starts from niche cluster 2, following niche cluster 1, 5, 0, 3, 4, and which is the same for this sample. And because on tracker it's trained on the multiple samples, so it will build a uniform trajectory for its all samples. And from this plot, we can know the spatial relationship between each niche clusters, and we can build the niche trajectory through these features. That we can realize the connectivity of this niche cluster. That we can see a clear path from 2, 1, 5, 0, 3, 4, which is consistent with the spatial plot. And the next thing is that we realize this anti-score. For anti-score, you can see that here this is a boundary, and which is going to the deeper. But somebody may want to say that, hey, I want to the anti-score start from boundary, not from these deep layers. So we can quickly reverse this one by just using 1 minus this anti-score, because this anti-score is already normalized from 0 to 1. So just using 1 minus this score, we can get a reverse version of this anti-score that starts from the boundary of the brain and going to the deep layers. And the next things we could know is that how about the cell type distribution along this anti-score. So we can use this function plot and cell type to anti-score, and we can see that along the anti-score, the VRMC, which marks the boundary of the brain, listed at the earliest positions, followed by other cell types, and especially for this L23 IT neurons, L45 IT neurons, L5 IT neurons, and L6 IT neurons. And in the end, we have the L6B IT neurons. And for other cells, they all have their specific distribution here, and actually they also have some cells which is separated to all the anti-scores here. And the last thing that is if we want to know in each anti-score which cell type composition for them, we can use this plot cell type composition in anti-score. And you can see that at the start point, that's each cluster 2, the main revision to it is we already see that's a cell type in the brain boundary. And then followed by niche class 1, which is L23 neurons, follows niche class 5 with L4 5 neurons, and niche class 0 with L5 neurons. And following niche class 3, which is L6 neurons. And in the end, it's the oligodendrocyte cells. So so that's all we can get from the contract, and you can see all the session info here. So take a summary. The on-track is a software to build a niche trajectory for all the cells in the sample. They can run on single samples or multiple samples. So I think that's all I shared today. And do you have any questions here?